Hello, everyone, and welcome to the Precision Medicine and Oncology virtual event. I am Sydney McNeil, and I will be your moderator for today's event. Before we get started today, we would like to share an informational video about Diaceutics, who is the sponsor of today's event. Right. Today's event will consist of a total of 5 informative presentations, as well as 2 breakout sessions with several of our speakers, which will take place in the networking lounge. During the breaks, we encourage you to visit the DXRX booth in the exhibit hall and diaceutics.com for more information. Now let's get started with today's first webinar, Implementation of Liquid Biopsy in Precision Oncology. Today's webinar is brought to you by Diaceutics. To learn more, visit diaceutics.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have any trouble seeing or hearing the presentation. I would now like to welcome our first speaker, Professor Dr. Klaus Pantel. For a complete biography on our speaker, please visit the presenter window at the top of your screen. Professor Dr. Klaus Pantel, you may now begin your presentation.
Hello, it's really my pleasure to talk today about liquid biopsy and the road from discovery to clinical implementation. The liquid biopsy concept is defined as the analysis of tumor cells, circulating tumor cells or their products, in particular circulating DNA, but also microRNAs or extracellular vesicles. And we have now also included the host cells, for example, circulating immune cells or endothelial cells in the blood or other body fluids. We have introduced this name in 2010 together with Catherine Alex Panabier from Montpellier, and the name liquid biopsy has really emerged in thousands of publications now. The rationale is very simple. Tissue biopsies are the gold standard in cancer diagnostics, but they are invasive and some locations are difficult to access. And even if you can assess a single biopsy with a metastatic lesion, it may relevant tumor clones that are relevant in other metastasis sites. So it does not actually include the intrapatient tumor heterogeneity. In contrast, the blood is a pool of tumor cells and their products released from primary and metastatic lesions, including in principle all locations in a cancer patient. And moreover, sequential tissue biopsies can be done, but they are really cumbersome and tedious and not very well accepted. But sequential blood analysis, of course, are more easier to do and more accepted by the patients and the clinicians. So this can also help to actually perform a surveillance after initial cancer therapy of what's going on with the cancer patient and early detection of relapse or minimal residual disease months before imaging is able to do that. So the vision is to get a more comprehensive and real-time tumor information by the analysis of blood or other blood body fluids. This concept of tracking cancer liquid biopsy was highlighted as a key discovery of the past 20 years in the latest Nature Milestone Cancer edition by Diana Romero. Well, where do the liquid biopsy analytes come from? In the early stages of tumor development, the primary tumor is the key source of circulating tumor cells and tumor cell products. Later on, the lymph node metastases contribute, and in the advanced stages, the distant metastases contribute to the pool of tumor cell and tumor cell products, particularly, for example, metastases in the bone, in the liver, or in the brain, or other sites of metastases. What can we measure in the blood? Well, we have the cellular fraction where we find the circulating tumor cells as single cells or clusters. We also find circulating immune cells, endothelial cells, or cancer-associated fibroblasts. And in the fluidic fraction, the plasma or serum, we can find the extracellular vesicles, proteins, RNAs, DNA, and also tumor-educated platelets. The two main analytes that have been used in many clinical studies today are the ctDNA and the CTCs. For the ctDNA, there are two principal types of analysis that we can do. We can actually take a hotspot gene panel that comprises the main genetic events of a given tumor type and then look for circulating DNA fragments in the blood. Or we can sequence the individual tumor and we can then perform an analysis with the individualized probes and follow up that in the blood. For the circulating tumor cells, the CTCs, we can use an enrichment of these cells, then a single cell detection and downstream molecular and functional characterization. And we can do that at the DNA, RNA, and protein level. And we have also developed cell line models for functional analyses. One of those analysis of circulating tumor cells is shown here as an example. This is an analysis of blood from patients with melanomas. We have done an enrichment and single cell detection here as a first step. Then we have done a single cell CTC isolation. And then we have looked at the DNA of these single CTCs and detected genetic aberrations relevant to clinical treatment, for example, BRAF mutations in these melanoma patients. And of course, this analysis can also give you an idea about the intra-patient heterogeneity. So not all the circulating tumor cells, not all the tumor cells are the same in a given patient. There's heterogeneity, and that might be also important for treatment options. We have also developed cell line models here, an international collaboration with many partners all over the world, where we have actually developed a cell line model from circulating breast cancer cells, which also have tumorigenic and metastatic capacity, and we also use that for testing drugs. In order to actually implement one of these discoveries into the benefit for the patient, which means the clinical implementation, we have established the European Liquid Biopsy Society as a consortium of more than 80 institutions from academia and industry. We have 4 working groups: dissemination education, clinical working group asking the right clinical questions, technology working group that probably can help to ask these questions and really address these questions, and then the regulatory working group which deals with the question how to get this reimbursed and implemented. And if you're interested in that consortium, we are open for new members. Just drop me or Claudia Koch a mail. We are also interested, of course, and active in guideline, and these are the guidelines from the European Society of Medical Oncology. I was part of that on the circulating tumor DNA assays, and I think it's very important to do the harmonization of methods and give guidelines for their clinical use. What are the clinical applications of liquid biopsy analysis? Well, first we have cancer screening and early detection of cancer. Then once cancer is detected, pre or post-operative risk assessments and prognosis. Then during the surveillance and follow-up period, the detection of early recurrence or minimal residual disease. Then the treatment selection, what kind of treatment is the best for the individual patients. Then using the blood analysis for assessing treatment responses and identifying potential resistant mechanisms. Let me give you a few examples, and I apologize for all the wonderful work that I cannot present today because of the time restrictions. For early detection of cancer, the circulating cell-free DNA is one of the key targets, and this DNA is released mainly from apoptotic tumor cells, but it can be also actively secreted through exosomes. And then this DNA is diluted in a sea of normal circulating DNA, and we have to develop and use very sensitive method to pick up this small fraction of tumor-derived cell-free DNA in the sea of normal DNA. What are the limitations? Well, we can have cancer-associated mutations in leukocytes, which are not derived from the solid tumor, and we call them chip mutations, and they have to be actually subtracted to avoid false positive findings. And some tumor lesions are non-shedders or very low shedders, which may lead to false negative findings, and we are still working on the biological reasons for this heterogeneity. For early cancer detection, there are many different assays that have been used. One of the assays is actually using methylation signatures on cell-free DNA and develop a multi-cancer detection test for screening of populations. And this was an initial publication a few years ago, showing actually the sensitivities at different tumor stages in different tumor types, from left to right, early or late stages. And you can see that for some tumor types, the sensitivity is quite good, but for other tumor types, such as breast cancer, for example, stage 1 and stage 2 tumors are not very well detected. This was then, of course, a follow-up publication published last year that actually showed on a very higher number of individuals analyzed that many cancer types can be detected with such screening tests, where screening tests do not exist today, that nearly half of the diagnosed cancers were identified as an early stage and half at a later stage. And of course, it shows that multi-cancer early detection seems to be feasible, but further research, larger trials with longer follow-up are needed. And this shows just the range of tumors that has been detected with this assay: hematologic malignancy and solid malignancy. And again, about half of the tumors were detected at an early stage 1 or 2. And this is, of course, very important because for early cancer screening, we want to detect the tumors as long as they are small, so they might be cured by surgery. So detecting early stages is really the key for cancer screening tests. Another approach is not using DNA, but using proteins, and there are many excellent papers published. This is from our own group. We have used a circulating stress-related protein, the CR61 protein, and detected it in the plasma of patients with cancer. These graphs show you the results of breast cancer. We have a difference between breast cancer samples and healthy controls. We can also detect early stages of breast cancer, and we have a sensitivity of above 70% at 100% specificity, which might be a nice assay to add on to other markers to actually improve early detection of breast cancer in the future. We also have analyzed this marker in lung cancer and asbestos-related diseases, so it seems to be not only restricted to breast cancer, but of course further research is needed before it can be implemented. Another approach is to combine different markers. We talk about multi-omics liquid biopsy, and here I think we can combine different markers, and one example for that is shown in the next slide. We have started a European network program with 17 partners all over Europe and UK and Israel. It's called PANC8. The goal is early detection of pancreatic cancer because it's a very deadly tumor and we want to detect it earlier to probably save a few more lives. And what we have are biobanks from patients with early stage pancreatic cancer and patients at risk to develop pancreatic cancer. Then we perform a whole range of liquid biopsy analysis from DNA, cells, proteins, metabolite, and hopefully using AI-driven bioinformatics, we can develop a classifier that detects this deadly tumor earlier than today. And we have also included health economy evaluation experts, and we have a patient organization, PCE, involved in it. So it's a very nice consortium tackling this very ambitious goal. Just to give you an idea how much information we can get, this was an analysis that we did in lung cancer blood. We actually enriched the blood for extracellular vesicles. Then together with our mass spec facility, we could find more than 5,000 proteins in 1.5 ml blood and actually categorize them to different functional groups. So you can imagine if you do that analysis, you can get an enormous amount of data, and you need really AI-driven bioinformatics to find out which are the appropriate candidate genes. The second application is risk assessment in early stage cancer. I mean, patients that have a higher risk may need more aggressive therapy. We have done this with the circulating tumor cell detection at primary diagnosis and could show that it's related to metastatic relapse and unfavorable overall survival in different solid tumor entities, particularly in breast cancer, but also others. And I think for the circulating DNA, this paper was published 1.5 years ago by Jin T and colleagues, and it addresses the question whether patients with stage 2 colon cancer need adjuvant therapy when they are negative for circulating tumor cells as liquid biopsy markers. So can we de-escalate these patients? And the result was very promising. There's also studies also now going on, and it shows that if these patients were negative for ctDNA or ctDNA guided, that they had inferior outcome. So maybe that is good news for the patients in terms of de-escalating also toxicity. But the study is still going on, and the observation period is now 5 years. The other application is surveillance of MRD and early detection of relapse. I think this is a very interesting application. Here, for example, one publication, they looked in the blood of breast cancer patients 8 years after initial diagnosis. And in these patients, about 10% of these patients had a positive ctDNA result 8 years later. And I think that they could show with a lead time of around 12 months before imaging that a relapse was actually coming. And it shows really the potential to really do even a long-term surveillance of breast cancer patients. And that is probably the beauty of blood analysis over time. We can detect minimal residual disease or early relapse before imaging. We are much more sensitive, so that may give us a window of opportunity, and it may also open a window of opportunity for clinical trials in this post-adjuvant actually phase of the disease development. And we can probably do some now clinical trials in that MRD window, which hopefully helps to prevent full-blown metastatic relapse. We have also another European consortium addressing this question, coordinated here together with Klaus Andersen from Aarhus, and the FPR lead is BMS. It's a private-public consortium with 22 partners. The objectives are to create reference standards and benchmark ctDNA assay. I mean, this is super important, harmonization of technologies. Then a robust clinical validation of ctDNA-based MRD in 3 model tumors, colorectal, pancreatic, and lung cancer, and then integrating MRD as a decision tool to guide treatment decisions. Liquid biopsy in advanced cancer patients is, of course, a very interesting application, which is probably close to clinical implementation. Here, what we have the advantage that we have higher concentration of circulating biomarkers, which makes our life a bit easier from the technical point of view. We can monitor tumor burden and response to therapy, and we are developing criteria to implement that in a new version of the RECIST. And we can detect alterations relevant to therapy targets or resistant mechanisms, which may help to say what therapy will work in an individual patient. Just to give you an idea, of course, this was a publication already some years ago where we had a lot of genomic alterations that are relevant to therapy, and this list is much longer today. But there are also transcript and protein analysis can help us to see what kind of targets or resistant mechanisms are expressed, for example, expression of HER2 or expression of PD-L1 in the context of immunotherapies. And this is really a very, of course, interesting field. Immunotherapy has revolutionized the treatment in oncology. We are focusing, of course, on immune checkpoint inhibition therapy in lung cancer and melanoma, but extend that also to other tumors. We monitor actually the tumor component here in the blood by monitoring the DNA and the CTC concentration and the molecular characteristics. But we also monitor the host, which I think is probably very important also in terms of response to immunotherapy. And we had one publication where we could show that actually that the patients that are treated with immunotherapy develop resistance with an influence of the thromboembolic system, and from Willebrand factor levels were good indicators for that. What we are doing actually here is in collaboration with an international network, the CCII network, Cancer Immunotherapy and Biology, established by the Nobel Prize laureate Tatsuko Honjo. Just to give you a couple of data regarding this aspect, here we have looked at the PD-L1 expression on CTCs. PD-L1, of course, a target for immunotherapy, prominent target. And we have seen that in this non-small cell lung cancer cohort, which is a real-world cohort in collaboration with a group of Martin Reck and Gus Hansdorf, in around 40% of the patients, we found circulating tumor cells. And in around 78% of these circulating tumor cells expressed the PD-L1 as a potential target of immunotherapy. However, what was very interesting, the situation was not black and white. We also saw some considerable intra-patient heterogeneity. Here you see the orange or red cell, that is a tumor cell detected by keratin staining. And in the first line A, it's co-expressing the PD-L1 seen by the blue stain, but in the line below that you have four cancer cells, one cluster, one single cell, and they are all PD-L1 negative. The next one, line C, is a single cell that is negative, And then in line D, you have again 2 cells that are positive and 1 that is weakly positive. So you see there is intra-patient heterogeneity, and that might be also an important mechanism of escape from therapy. Together with our colleagues at the Dermatology department, Professor Christopher Geppert and Stefan Schneider and colleagues, we have also looked into the use of a very sensitive technology looking at ctDNA in patients with metastatic melanoma that receive immune checkpoint inhibition therapy. And if you use this very sensitive assay, which can go down to 1 parts per million, we can detect a whole variety of melanoma genes, not only the BRAF gene or NRAS gene, which are frequent, but also others. So we get much more information. And this information seems to be also clinically relevant, because if you look at the Kaplan-Meier curve on the right side, then the patients where they have ctDNA clearance, they had a good, survival compared to the patients that had no clearance. So it seems to be also important to have sensitive assays even in the metastatic setting. Just to give you an idea about the host, we should not forget about the host. Each patient may have different capabilities to actually respond to immunotherapy. And here we measured the von Willebrand factor as an indicator of the thrombotic system in the patients. And we could show that the patients that had primary resistance compared to primary response had a higher level, a higher activation of the thrombotic system. And that also was relevant for progression-free and overall survival. So the host is also important. At the end, I think I want to stress that in order to bring liquid biopsy tests into clinical practice, we need interventional clinical studies showing clinical utility. And one of these studies was published on the CTCs lately with the French group, a multi-center French interventional trial, which actually compared the clinical decision guidelines to treat patients with metastatic H-positive breast cancer compared to the recommendations made by the CTC counts. And they could show that in 25% of the patients, the recommendation was that the risk is low, but the CTC indicated a higher risk. And if the patients got a more aggressive therapy, the chemotherapy, they had a benefit in survival. And in the second group, it was in around 14% of the patients, the clinical guidelines indicated more aggressive disease, but the CTC count less aggressive disease. And if these patients were treated with a less aggressive therapy, it was not the outcome was not inferior. We did another study at a multi-center interventional trial in Germany, actually analyzing the HER2 status on circulating tumor cells. We could already show years ago, and other groups too, that there was a discordance between the primary status of HER2 and the CTCs at the time of metastatic relapse. So there seemed to be a shift, a subtype shift from HER2 negative to HER2 positive in some of these patients. And we were interested whether these patients will actually benefit from treatment with an anti-HER2 therapy. And in this DETECT-3 trial, these patients received an anti-HER2 therapy with lapatinib. And these patients had initially HER2 negative tumors, but the HER2 positive CTCs at the time of metastatic relapse. So we were interested, do the patients benefit from such a therapy? And this is a result published now recently in terms of the overall survival. We see here that the addition of anti-HER2 therapy to standard therapy had an overall survival advantage that was significant. And it's the first interventional study indicating clinical utility of CTC characterization. Of course, it needs to be confirmed by larger trials, but I think it's a very promising result. So if you're more interested in liquid biopsy, we have published a journal, a special issue in clinical chemistry with many good articles on liquid biopsy analysis. There's also one article of our consortium focusing on cancer or of unknown primary, COP, which I think is a very interesting, cancer entity or group of cancer entities to assess with liquid biopsy. And if you're interested in that, I can really recommend to read this edition. So in conclusion, early detection of primary cancer and MRD and relapse requires very sensitive assays. Tumor burden molecular characterization of alterations relevant to systemic therapy can be monitored in advanced cancer patients. That may lead to a new RECIST version, and we are working on it, and also other groups to complement the imaging. Then the assays need to be validated and harmonized, and we need quality assurance measures. We need, of course, technical validation independent, and the ELBS is helpful in that regard. Then the combination of different liquid biopsy markers may lead to higher accuracy. So we may not focus just on one type of marker, but we may combine markers to get more information. Then the resistance to immunotherapy may depend on both tumor and host responses. Immunology is a very complex field, and the biology is super complex, and it may not be enough to just measure the tumor response, but also the host response should be included. And last but not least, interventional studies are important to demonstrate clinical utility of liquid biopsy, and there are many interventional studies going on, also with the circulating tumor DNA. I just had a chance to show a few of our own studies, but many groups are working in that direction, and also including new markers such as EVs and others. So this is really very important to really define an intervention based on your blood test results and then do the clinical trial for that. So at the end, I would like to thank the Liquid Biopsy Research Network here in Hamburg at the University Medical Center, which consists of institutes, research institutes and clinics treating cancer patients. I would like also to thank my team at the Institute of Tumor Biology here at the Center of Experimental Medicine and University Comprehensive Cancer Center, Hamburg. And in particular, I like to thank the actually funding organization at the European level, the ERC grants that are received, but also the IMI and Horizon grants and Transcan grants, which help to do a lot of good research. And in particular, also I like to thank the German funding agencies, the GFG, the BMBF, and Deutsche Krebshilfe, and many others that support our work here in Hamburg. If you are more interested in liquid biopsy, there is an interesting conference in November in San Diego, organized by the AACR, and I think this will be 3.5 days of hot research results presented in this beautiful location. And if you don't want to travel to California, you may want to go to the south of France next year. So we have our 14th conference on minimal residual cancer and liquid biopsy, and it's really comprising all kinds of applications in liquid biopsy, not only MRC. And we have a wonderful list of speakers here that all agreed to come to the south of France. And so that might be another good chance to get updated information on liquid biopsy. Thank you very much for your attention, and I'm also happy to address and answer your questions.
All right, wonderful. Thank you, Professor Dr. Klaus Pantel, for your very informative presentation. We will now go ahead and move into the Q&A portion of the webinar. If you happen to have a question that you would like to ask, please do so now. Just click on the Ask a Question box at the bottom, or I'm sorry, at the far left of your screen, and we will answer as many as we have time for. Okay, let's go ahead and get started. The first question that we have here says, what is preventing or slowing down the adoption of liquid biopsy for monitoring cancer recurrence in many countries?
Well, I think that we have in many countries a very complex system of implementing new diagnostic tests, into the reimbursement scheme. And I think that this is, of course, very important to find good roadmaps for what clinical applications liquid biopsy can be really implemented into reimbursement schemes. And that's, of course, one of the big bottlenecks for all new diagnostic measures. The other thing is that we need also more, I would say, quality assurance and harmonization of methods. There are many, for example, ctDNA or CTC assays that are available in the literature, and sometimes it's quite confusing for people to say which one we should really choose. And I think this kind of quality assurance is also a very important point to make sure that the assays that are used for clinical decision making in the future are also reliable and reproducible in different laboratories.
All right, wonderful. Thank you. We have another here. It says, what are the current limits of liquid biopsy in terms of false positive and false negative results, and in terms of MRD? Does ctDNA represent a clinical, feasible, and sensitive approach for residual tumor tracking?
Yeah, this is actually another good question that we are addressing in our guide MRD EU consortium at present. At present, we have seen that if the ctDNA is positive, it seems to be correlated with later relapse. And if it's negative, it has a lesser correlation to relapse, so it's a prognostic factor. However, the negative predictive value is not optimal, and we try to see whether we can identify more patients with MRD that later develop relapse, so to improve the negative predictive value. But if we also use more and more sensitive assays, we may also come into a specificity problem, because aging individuals that actually carry a lot of genomic aberrations in their leukocytes or also in other cells. So there might be also a quantitative cutoff if we have super sensitive assays in order to avoid false positive findings. But right now we are focusing more on the false negative findings with regard to MRD detection. But then I think this is a wonderful field for future clinical trials, because then if the next question is, once we can detect MRD reliably, the question is, of course, what kind of therapy can block the progression of MRD to full-blown metastasis? And this question can be, of course, addressed then in clinical trials.
All right, wonderful. It looks like we have time for a few more here. This next question states, standardization of LBX test around Europe and the globe. Where are we at the moment?
Well, I think there are several groups that are working on standardization all over the world. We also have an alliance on standardization, which is shared by the foundation of the NIH. We have regular meetings with the different groups all over the world that are doing that, like ELBS, Friends of Cancer Research, or Blood Bank. And I think we are trying to develop common guidelines to make sure that the quality assurance is implemented together with the clinical implementation of these assays. And I think this is super important. Imagine if you say that a negative ctDNA test may lead to de-escalation of therapy. You really want to be sure that this is a standardized assays and that you have a good quality assurance to make sure that this important result is really reliable and reproducible. So I guess that these activities also of our European Liquid Biopsy Society are super important also for the clinical implementation. Mm-hmm. And we have also performed ring experiments in the ELBS, and we compared different technologies and see how reproducible different laboratories are. So these are activities in the ELBS which we will continue to do.
Perfect. Thank you so much. Here is another. How many years will it take for implementation of liquid biopsy testing in routine diagnostic?
Yeah, that's a good question. I mean, prediction of the future is usually very difficult, right? And but I see already the first tests that are now being implemented in different countries. Particularly in patients with advanced disease when there is no tissue available. We have already recommended that in our ESMO recommendation that then the blood analysis might be another choice to get information on the individual molecular characteristics of the tumor, and that may help to steer therapy, for example, in lung cancer, where we have certain molecular alterations that guide really therapy very well. And I think these could be the first applications that are also implemented. And I would say we already have the foot in the door here. And other applications will come. For example, ESR1 mutations in breast cancer also seem to be very relevant and can be also assessed nicely with ctDNA. And maybe HER2 for the CTCs could become interesting, particularly in the HER2 low therapy setting. So I would say within the next 5 years, I assume we will have a series of specific clinical applications as companion diagnostic to therapy, but also, for example, as a method to escalate or de-escalate therapies.
Perfect. Thank you so much. Okay, it looks like we have time for one more here. And this one comes in as a follow-up question with regards to reimbursement. Can you comment on cost effectiveness of liquid biopsy testing? And more specifically, how do payers feel about currently available assays used in surveillance that have significant costs?
Well, this is, of course, an issue that we are actually dealing with our health economy experts in our EU consortia. That's why we have actually implemented health economy experts for early detection of cancer, but also early detection of MRD. This really depends probably how many tests are necessary to detect MRD. If it's, for example, just 2 tests or 1 or 2 tests, and then that's probably a good thing to do. If you have to test the patient every 3 months for 10 years, then the cost would probably increase. So it really depends on what kind of scheme is sufficient to detect MRD. But I would also say that if you can really avoid full-blown metastasis by actually avoiding the progression of MRD to metastatic disease, then you may save a lot of costs because these patients then can go to work and they may not need all the therapies that are necessary for end-stage disease. So I'm not an economist, but I would think that even a test which is $500 or $1,000 or $2,000 could be still economically efficient compared to the very high treatment costs that may be avoided by curing a patient at the MRD stage.
Perfect. Thank you. Well, it looks like that is all of the questions that we have for today. Thank you again, Professor Dr. Klaus Pantel, for your time today and for your very important focus on this area of precision medicine. We would also like to thank our sponsor, Diaceutics, for underwriting today's webcast. Before we go, I would also like to thank the audience for joining us today. As a reminder, this webcast can be viewed on demand. We will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.